You've really listened to three eminent experts in the field. The floor is now yours. Any questions that you may have to any of the experts. If you have a question, please just say your name, your affiliation, and then maybe also say which one of the three experts you're asking the question to. Yes.
My name is Peter Kolominski. I'm professor of neurology at University of Erlangen in Germany. I have a comment and a question. First, the comment. It was said that EPAT is something new because industry and researchers are collaborating together. I do not agree with this because we have, since decades in Europe, a very intense collaboration between scientific research group like the European Research Council and industry. What is new is that industry companies are working together. This is new. And as Simon Lovestown presented this morning, big trials are very expensive and they are very risky. And the cooperation of industry in EPAR is a risk minimization strategy so you can decrease costs in research and development. So far my comment. My question to Serge. I think that the adaptive trial design is something innovative, and this leads me to the question: you want to bring biomarkers and drugs very quick to the patients. And what I am missing in EPAT is the health technology assessment competence. We have in every European jurisdiction health technology agencies like IQWiC in Germany, NICE in UK, HAS in France, and these bodies are deciding whether these drugs and biomarkers will be reimbursed or not. And I am really surprised that you do not have implemented such a, let's say, competence. We have this competence already on the European level, like the UNETA network. And please, could you comment on this?
Yeah, and it's a fair comment. I think we had when we started out building the consortium, we discussed having the health technology assessment agencies involved from the very beginning. And I can assure you it's part of what we are going to do. Where we struggled with, and we had a debate with it with the European Medicines Agency, is some of the things we do or some of the approaches we are going to use are, without exaggerating, trying to exaggerate it, some of them are so new, we are still struggling with them. So the way we're going to approach this is get in a conversation with the European Medicines Agency, the Food and Drug Administration first, kind of see that we're kind of on a level plane there, and then in collaboration with, in Europe, then the European Medicines Agency, pull in the Health Technology Assessment Agency. So it's not something we, let's say, we deliberately did not pull them in from the very beginning, but it's also not something we kind of said we're not going to involve them. It's more a staged approach, because I think for some of these things, we're still trying to find our footing. I think it's kind of, I hope that answers your question.
We have another question up here, or?
Thank you. I'm Dave Morgan from the University of South Florida. I'm one of those Americans you were talking about. So we do have a little bit of experience in the US with prevention studies, and my site is one of those that's running the A4 clinical trial. And we've run into a pretty significant problem, which is a much smaller fraction of the people that we screen end up being amyloid positive than we expected. And when I look at the clinical trial data sets, I'm always awed at how educated everybody who volunteers for clinical trials really is. In the US, I think it's about 15 years of education, which means they've basically all graduated from college. And we know that the risk for AD is much greater in those people who are having to work hourly jobs, who didn't complete high school or have a high school education, and they probably don't have the lives of leisure that people like Simon and myself have to take off a day, to come in to do all of these probings and proddings that we're doing to find out what's really wrong with them. So the question I have is, do you have a mechanism by which to include in this recruitment those people who aren't necessarily coming to you and volunteering, but are you using this randomized group that you talked about this morning to try and get a much more representative sample? And again, emphasizing that point, the right patients.
That's a question to Simon.
It's a really good point, Dave. And I think the short answer is we don't have any magic way of doing this, and it's very difficult for all of us. What I think we have an opportunity to is by building on the parent cohorts that have literally millions of people all over Europe, we have at least a chance of being able to have a more representative group of individuals. But the primary problem that you're quite rightly pointing out, that the people in those parent cohorts are themselves not entirely typical of the...
Low risk.
Yeah, exactly. And that is going to remain a problem. What I hope we'll be able to do is, because we've got a much bigger set, because we're going to these millions of original people, we might be able to end up with a more representative group than if we just are the primary recruiters, if you see what I mean. So that's one thing. Second thing is, I really want us to, and I don't think this is a job that's completed by any means, but I really want us to build on the algorithms for risk. So we're obviously speaking to A4, Ray's Paul, to try and take the learnings that you have from those algorithms. So for other people in the audience, what I'm talking about here is, can we take some data such as cognition, combined with ApoE, combined with perhaps education, combined with perhaps stratification by other factors such as employment or something, to find a group of people who are at particularly high risk of having the primary condition that we're interested in here, which is preclinical biomarker positive AD. So we have an opportunity to both implement existing algorithms like those that you're using, try and tweak those to make them better, and then the adaptive process to learn from those algorithms to make them better in the second and subsequent waves of the study.
I also saw another question down here from, where's the mic gone?
I can speak loud. Can everybody hear me? Yes. Thanks for the presentation. I think this is a very interesting initiative and program. So my question is, what are the timelines for that? So you talk about recruiting people very fast and good outcomes, but I'm missing timelines. And the other question is regarding your comments. I would like to make an additional comment. At the end, it is important that patients get access to those results and outcomes. We have a lot of experience that we invest in R&D, and then the translation to the clinical practice is a difficulty, because the authorities say, you don't meet the endpoints we want to see, and at the end they don't pay for that. I strongly would recommend to involve HTA bodies or even appraisal bodies like GBA in a really early process step. To my person, my name is Hedwig Siliet. I'm working for Piramal Imaging, a diagnostic company. We develop tracers, first tracer in the market, and we are working on translation from R&D and clinical practice. So.
And it seems that we have two volunteers to answer that question. Serge,
we can
and then
both
Simon.
answer. So to your first point, timelines. The cohort study, the aim is to get it up and running with the first research participant in by April next year. The second part, the actual proof of concept study, that's a slightly more complicated thing. The goal is to start in September next year. Now, the reason I say it's a slightly more complicated thing is what we do in EPAD with all the pharmaceutical companies involved. And maybe I should already stress, although initially it's probably going to be pharmaceutical companies providing compounds. We set out in the beginning, EPAD is not limited to pharmaceutical treatments. We are open to many other approaches if they make scientific sense. It's not a free-for-all, but we are open to discussing it. So what we are currently doing is building that virtual pipeline so that we can actually, because as Simon said, we are building a 6,000 participant cohort. We're not going to do that overnight, so we're going to need to stage who we're going to be able to take into the trial. The one caveat there is drug development is not without its risk. We have a pretty good view on, let's say, the first 2 or 3 compounds that might come in. But something something can always go wrong between now and September. We might run into a toxicology finding which takes the compound out of the. So there is some risk there. I don't know if you want to.
I think that we already, unfortunately, have run out of time because the next session is going to start in about 2 minutes. So I really want to thank all 3 speakers about this really interesting update on an absolutely fantastic and exciting project. I just wanted to repeat what Simon said in his introduction. If we continue to do business as usual, we're going to risk failures just as usual. We will be continuing to have 101 trials with 3 success rates. And ultimately, this project is trying to make sure that research in the future will be better for patients, it will be better for companies, but all in all, it will be better for society. So thank you to all 3 speakers for their dedication, and we look forward to updates in the future about this project.